Yeah, so thanks for this opportunity again. Today I'm going to talk to you about how we use deep learning models for decoding regulatory DNA and non-coding genetic variation. We're going to focus on these kinds of regulatory profiling assays, where the goal is to use experiments to get genome-wide profiles of various kinds of biochemical activity. In this case, I'm showing you right here, ChIP-seq experiments targeting a variety of different transcription factors. And here's some attack-seq or DNA-seq experiments that allow you to measure chromatin accessibility genome-wide. And there are many different variations of these: DNA-seq, now the single-cell attack-seq, there's PRO-seq, PRO-cap. So we have a huge collection of different kinds of molecular profiling assays now, and they're allowing us to profile bulk tissues as well as individual cells at incredible resolutions and across vast cell states and cell types. The question that we're really interested in is how DNA sequence encodes this sort of molecular activity, and hopefully as a surrogate for function. So we want to try to use models that will allow us to decipher the DNA sequence basis of this activity. So we're going to sort of twist this problem into a classical machine learning task and a supervised machine learning task, where you're given some sequence across the entire genome, 3 billion base pairs, and you're given some experimental profile from some experiment, and you want to learn a mapping from sequence to this sort of profile. So typically, the strategy people have used is they will take this kind of data. You will often binarize it. So you will, for example, run a peak caller or something, and you'll say, okay, here's a peak, here's a peak. There's 4 peaks. The rest of it is background. So you'll binarize the genome, and then you will take bins of about 500 to 1,000 base pairs of sequence, and you'll have binary labels. Of course, you can also replace those binary labels with actual continuous, say, enrichment scores of sorts. But basically what's happening is the sequence is being summarized to a single scalar typically. And then you're learning some kind of model that can map sequences to these scalar labels. And there's massive work in this space, for decades. But what has happened more recently is the assays have been getting more and more interesting and more high resolution. So for example, if you look at DNA-seq and attack-seq data, if you take typically processed data sets, you'll see stuff like this. You'll see kind of these nice peaks and valleys, and the resolution of these peaks is about 200 base pairs. But if you actually zoom into any of these peaks, you see what's happening at the read coverage profiles at single base pair resolution. With deep enough data, you can often see really interesting things going on. So for example, proteins that bind DNA will protect the DNA from enzymatic digestion, and so they will create what are called often footprints. So you'll see a lack of coverage right where the protein binds. And it's not just that, but right around that footprint where the protein binds, there's a specific architecture of read coverage, and that really quite nicely reflects the protein DNA interface. So it's worth taking advantage of this sort of structure of reads in the data rather than just summarizing all this beautiful information into one scalar value capturing coverage in that bin, right? So we want to kind of really move into single base pair resolution models. It's difficult because data is very noisy, right? So that's probably why people have stuck to these sort of binarized models or sort of summarized scalar models. It also gets difficult because we have to think about what really affects these kinds of footprint shapes and spans. There's a variety of biological entities. So there's obviously a residence time of transcription factors. That's how long they stay on DNA determines, for example, how deep these footprints are. So here are 2 transcription factors that have high residence times. They produce these beautiful deep footprints. Then you have other transcription factors that have low residence time, and they often produce shallow footprints. But there's many other factors. There's the affinity of the actual sequence that the TF is binding to. That can have an influence. There is not just the exact sequence it's binding to, but the surrounding context has a big impact. There's lots of cooperative effects. So overall syntax of the sequence matters. The concentration of transcription factors matter. And then there is the big elephant in the room, which is enzyme preference. So all the experiments we perform, we think that they are the ground truth, but they are all using various kinds of enzymatic tricks. And sometimes the enzymes can have pretty strong effects on the measurements you're looking at. And at least in prior work, people have shown that at least for attack-seq and DNA-seq experiments, enzymatic biases can be pretty strong. So, we were thinking of how we can learn these enzymatic biases, and people have built sort of simpler models in the past from a control experiment. So, for example, you can take naked DNA and just transpose it, or you can do a DNA-seq experiment on naked DNA, and then just sort of model the sequence preference of DNAs and ATAC-seq. But that doesn't account for, protocol-specific differences, batch-specific effects. So you're wondering if we can just learn this enzymatic preference just from the experiment itself. So one nice thing that we have is when you perform any kind of profiling experiment on the genome, most of the actual events you care about, like these binding sites, are sparse. So you have maybe 10,000 or 100,000 events where you actually see signal, and the rest of the genome is just sort of background. So the nice thing is we have actually a lot of background data. So why don't we just train some neural network on the background and see what it learns? That's exactly what we do. So instead of training models on the peak regions, which usually people care about, we just train a neural network on the background, basically regions that have, very sparse and crappy signal, like things you don't usually care about. Now, what's really interesting is if you train a model, a sequence model, a neural network that just takes sequence and tries to map it to base resolution coverage in background regions, you would have expected the model to have really crappy performance, because you would hope that background is just random noise and you can't learn anything. Actually, you can learn it extremely well. So we learn extremely predictive model. And actually, if you interpret this model, and the way we do that is we use two methods, deep-lift and Modisco. Deep-lift is a simple feature attribution method. So it takes any sequence in the genome, takes a model and can compute, base pair resolution importance scores for every sequence. And then this animation shows what Modisco does is takes those base pair resolution attribution profiles, identifies high importance words, and then clusters and collapses those words to learn sort of unified motifs. And when you do that, you see that, for at least for ATAC-seq, if you interpret this sort of what we call the background model, the bias model, you learn a collection of different motifs, really well-defined motifs. Many of these are, at least the first one that you see right here, which accounts for about 43% of the instances that you see in the genome. This is the classical TN5 motif that people have seen before when they construct position weight matrices. But it's more complicated. The story is more complicated. It's not just one motif, a single position weight matrix that is fully capturing the sequence preference of TN5. There's actually a variety of different other variations of this motif that the neural network is effectively learning and combining in complex nonlinear ways. So we can kind of interpret the model and try to understand, okay, it's not only learning the canonical TN5 motif, it's learning a whole other variety of variations of sequence affinity. So this is very interesting because now we have a model that claims that you can predict background with incredibly high accuracy, and we can actually, in fact, pull out what the model's learning. So now, if you want to actually train a model on the entire genome, which includes the peak regions, we need to be careful, because if we're not careful, we will not only learn the underlying transcription factor biology, but it will be heavily confounded with all of these sequence biases. So what we do is we just sort of design a new a bias factorized model. It's very simple, actually. It's a simple convolutional neural network. Again, there are two sort of paths to get to the output. The goal is to get from DNA sequence to base pair resolution profiles, okay? Read counts at single base pair resolution. And we have two output heads for the model. One computes the sort of total coverage within, say, 1,000 base pair window, and the other component predicts how the total coverage, the total number of reads, are distributed at every base pair in the 1,000 base pair. So we have kind of a base pair resolution, probability profile, and then sort of the total coverage over the entire sequence. And to try to predict these two entities, we have two components of the model. One is this bias model that we pre-trained on the background, right? So we try to explain as much of the signal in any region of the genome as we can with the bias model that's pre-trained. So this model is frozen. And then this second component of the model is what we train on the entire genome. So it's trying to learn the residual that the frozen bias model doesn't learn. So it's super simple, right? It's like regressing out the bias and then fitting the residual through this secondary module. So let me show you what actually happens. All right. So this is the observed data at some region in the genome. This is the predicted data from the model if you do not do any bias correction. You can see it actually looks incredibly accurate at single base pair resolution. All the spikes, all the valleys, everything looks great. So you might be really happy and be done with it. Now, if you interpret this model, you see something interesting. You see a transcription factor motif right here. This is very well known. This is a CDCF motif, which drives chromatin accessibility pretty strongly. You can see this beautiful footprint, right? This valley of coverage. But what happens is you not only see the CDCF motif, but you see all this other garbage popping up, right? So the model is learning all kinds of importance for all these kinds of base pairs, which don't match anything we previously know. Now, remember, we have this bias model that we have pre-trained on the background. So we can take that bias model and predict what the bias might be in this peak region, right? And when we do that, this is what it looks like. So it's kind of shocking because you see that the predicted bias profile is very, very similar to the actual observed profile, except in this zone where the transcription factor binds. And if you interpret the bias model, you actually see these spurious bases that are high importance in the uncorrected model are exactly what the bias model picks up to. You can see that these regions are very similar. This region, where the transcription factor actually binds, is absent in the bias model, as you might expect. The background is not going to learn transcription factor motifs. But the joint model, the model that is just trained on the uncorrected model, that is just trained on the data as is, learns the transcription factor motifs as well as all this spurious signal. So now when we do the bias factorized correction, this is what we learned. This is the latent signal that the model predicts. It's actually quite beautiful. There is this hidden latent footprint that's sitting in there. You don't really see it in the observed data. It's kind of very noisy. And if you interpret the latent model, actually you pick up just the motif, really beautiful. And you pick up these weak repressors, the ZBTB7A repressors, which are invisible in the original model, right? And if you compare this to previous approaches that do kind of simple statistical bias correction, like there's a method called hint attack, there's another method called Tobias. If you train the models directly on those corrected data, you can see that they don't actually work as well. So they still have residual signal. They're not really correcting the bias as well as a neural network trained on background can. So the nice thing here is we are using the neural network's power to learn patterns extremely accurately to actually learn bias and then regress out the bias and then learn the residual and then interpret the residual. And that's what's giving us the clean biology. We get overall really nice performance. Our global correlations are quite nice at the level of total coverage. Also, if you looked at Jensen-Shannon distance between observed and predicted profiles, this distribution is what replicates look like, pseudo-replicates. This is what our predictions look like. So we're kind of overlapping pseudo-replicates. This is what the lower bounds would be randomized data versus like an average predictor, right? So we're actually doing, as I showed you in that cherry-picked example, the observed and predicted profiles really look like they're mimicking each other. We're getting very nice predictions overall. So if you interpret overall what motifs the model is learning, if you don't actually correct for biases, you not only, you sort of learn transcription factor motifs, you see a RUNX and IRF and SPI1 and so forth, but there's this giant TN5 monster sitting right here, and there's another version of it here. Many of the motifs are confounded by TN5 bias. You can see this huge GC kind of haze around this motif and so forth. And after you do bias correction, you get absolutely gorgeous, clean motifs that you can pull out of the data. Now you can actually use the model to study, transcription factor footprinting. So what that means is we can try to take individual motifs that we care about, and we can in silico insert them in synthetic sequences, and we can push them to the model, and the model can predict what the actual footprints for those transcription factors would look like. So here I'm showing you 4 transcription factors, which are expressed in different cell types. NF-kappa B is expressed in GM12878, which is an LCL, a lymphoblastoid cell line. GATA is expressed specifically in leukemia cell line, K562. HNF4A is specifically expressed in a liver carcinoma line. So these are very cell type specific transcription factors. Now, if you try to predict their footprints without bias correction, with a model trained on ATAC-seq data in these cell types, you see that irrespective of the cell type, you see the red profiles, those are uncorrected footprints. All the models just claim that there are footprints for these transcription factors, despite the fact that they're expressed only in specific cell types. After you do bias correction, you see this beautiful specificity, right? You can see the black curves. There's a beautiful footprint for NF-kappa B, only in LCLs, but it's a flat line in K562 and FG2. GATA1 shows this nice weak footprint. it's a low residence time TF, so it has weak footprint here, a flatlining here. HNF4A is really specific to HG2, and SP1, which is a sort of a control, it's expressed in all three cell types. You can see a nice footprint in all three, right? And so you can see the dramatic correction in the definition of a footprint itself. Like people typically think of these, these red profiles as the footprints, but they're actually mostly just bias, at least in ataxia. So correcting this bias is really useful and reveal very cell type specific biology. Now we can use the models to not only study the syntax of regulatory DNA, discovering these motifs, interpreting individual sequences, but we can also use them as oracles to predict effects of genetic variation. So here I'm showing you how we can use the models to predict effects of QTLs. So here's a positive control. This is a very well studied QTL in LCLs. It's a variant that affects binding of a particular transcription factor, SPY1. It also affects chromatin accessibility, and it affects histone modifications. It affects gene expression. It happens to also be a GWAS, fine map variant. And here what I'm showing you is we've trained three models on, one is on a transcription factor ChIP-seq dataset, the other's on a DNA-seq dataset, the other's on a histone ChIP-seq dataset. And what I'm showing you here are really predictions of the model. The observed data at single base per resolution, I can't even show it to you because it really is just like sparse garbage. What we're seeing really is very high resolution predictions of what the two alleles of this variant do. You can see this gorgeous amplification of the signal. And not just amplification of the signal, but you'll see that there is a shift in the valley of the profile. So the blue profile sort of has a valley right here, and the red profile has two valleys. It has a valley right here, and then another valley right here. And if you interpret the model and see what it's doing, the C allele, when the sequence of the C allele actually has a weak motif right here, that's what drives this blue profile, this blue valley right here. And when you switch the C to a G allele, it creates this really strong secondary motif. And these two motifs cooperate to actually generate this really strong profile. That's the reason you see this shift in the shape of the valley itself. This particular valley is coinciding with this motif. This really strong valley is corresponding with this motif. And you see consistent interpretations from the three models, three separate models trained on three separate readouts, tell you the same story. That is these two motifs that are driving effects. So what we've done is we've taken our models and compared systematically models predictions against various gold standard experiments. For example, here we're comparing against DNA-seq QTLs. These are, again, variants that affect variation in DNA-seq activity across a large collection of individuals. We're looking at precision recall curves. We're trying to classify, is a variant statistically significant DSQTL or not? And I'm just showing you comparisons of our models against two other strong baselines. One is a older generation model, the Delta SVM model. It's an SVM that's trained to classify peak regions from non-peak regions. And the precision recall curve is right here, the purple one. Then recently, DeepMind released this absolutely gigantic model called Informer. It's a multitask giant transformer trained on tens of thousands of readouts. That's this particular red and green curves. Okay, and so they substantially outperform most of the previous state-of-the-art methods, including SVMs. But here's our little Chrome BPNet model. This is trained on one experiment. It's a tiny model. You can train it on one GPU in 4 hours, and it substantially outperforms the Informer model by a big margin. We have other validation experiments, like, for example, we have compared our model's predictions, or here on the y-axis, against various chromatin accessibility QTLs from ataxic experiments in a large collection of African individuals in LCLs. We also compare it to allele-specific effects in those same cohorts. And here, the really interesting thing is we've trained our models on a reference ataxic data set from a European individual. And so we're training models on a reference data set from an individual from a different ancestry group and testing it on QTLs from very diverse African populations. And we see really nice concordance. This is pretty state-of-the-art compared to most other methods. And we see nice concordance between allelic imbalance as well as CAQTL betas. We've tested what happens to these models if we train them on, as I showed you, a European individual, or if we train the models actually on African individuals. Does that make a difference? The nice thing we find is that since the models are capturing what we think as regulatory biology, although the variants are quite different in terms of allele frequencies between populations, the predictions are very generalizable. So we see barely any drop in prediction between models trained on African individuals or European individuals or African subpopulations in terms of predicting QTL effects. So this is kind of a nice generalization that we see capability of the models even across ancestry groups. We can use this to find map a GWAS loci. So here's a nice example. This is a GWAS locus in multiple sclerosis. This is the IL-7 locus. So as you know, in GWAS, the problem is you identify the locus, but it's very hard to figure out what the causal variant in the locus is because of the LD that you see often in the locus. So this locus, I'm showing you a small collection of variants right here. These are all genetic variants. Here's the LD map for European individuals. You can see there's very strong LD between all these variants. So it's very hard to tell which one is a causal variant. If you look at the African LD map, you can actually see the story is a little bit better. You can start breaking apart the LD block. These two variants are in high LD, these two are in high LD, these are in high LD. So you're trying to reduce the potential number of candidates you might be looking at. If you look at EQTL data from Europeans, these variants have been highlighted as significant EQTLs. If you look at African EQTLs from African population, you actually see, again, a smaller subset, but a different subset being picked up. If you look at African CAQTLs, it pinpoints these ones. And so, you can already see that molecular data, QTL data is helping us kind of reduce the number of candidates to potentially look at. But the beautiful thing is if we just take our model trained on a reference individual, a reference data set from a European individual, and we score all these variants, we highlight basically one variant in the locus that has a really strong significant effect. And that happens to be exactly the variant that everything else converges on, all right? And you can interpret the model and gives you this really nice interpretation. These are the two alleles. You can see the orange curve and the blue curve, capturing the base resolution profiles predicted by the model for the two alleles. You can interpret the underlying sequence, and the story comes as, the C allele creates this OCT4 motif that cooperates with this SPI motif to create a strong enhancer. The A allele destroys the OCT4 motif while keeping the SPI1 motif intact. And you can see a cooperative effect where when you destroy the OCT motif, it also reduces the contribution scores of the SPI1 motif nearby, right? We've tested this also on, fine map GWAS variants from UK Biobank. And I won't spend too much time on this plot, but the key point to show is as the posterior probability confidence increases on the x-axis, the observed versus expected overlap of our predictions from our model with the fine map variants starts getting much, much higher. So you can see, you're getting like 60, 80, 100-fold enrichments as you go up the posterior probability threshold. So we are pretty confident that the models are now starting to pick up really nice effects. And one thing I didn't mention is this model is not even trained on the human genome. It's actually trained on vascular smooth muscle cells, ataxic data in mice. So this is really cool because we can also train models in other species where you have data from tissues or cell states which are hard to obtain from humans, and those models can still generalize quite nicely. I'll skip this. I think I've run out of time. Is that right? So I'll just skip this. But we can interpret again these variants and identify really nice cooperative effects between variants in the same haplotype. We can apply these kinds of models to also rare diseases. So we are looking at congenital heart disease and training models on single cell ataxic data from human fetal hearts. This we recently published just last year, and we can validate a lot of these effects through CRISPR experiments, through reporter assays, and also through phenotypic experiments, as shown right here. So with that, I'll just summarize. So I showed you we have these new Chrome BPNet models that are base pair resolution models of ataxic DNA-seq. They provide superior enzyme bias correction to previous methods. They can reveal predictive motifs, instances, footprints at base pair resolution. They can predict and interpret CAQTLs, GWAS loci, de novo mutations. It's a nice example of a small model really competing and outperforming a giant, foundation model of sorts. Our code is available as a beta release. Papers coming soon. And just a quick future outlook. I mentioned this in some of our discussions yesterday as well as today, but there's going to be an explosion of models over the next few years, and I think it's really important that we have sort of centralized repositories for models, just as we have for data. These model zoos must be linked back to the raw as well as processed data sets for provenance. Otherwise, you won't know what the models are actually trained on. So that's quite useless. Honestly, if you have a model and don't know what exactly it's trained on, you might as well not use a model at all. Similar to explosion of models, there are going to be explosion of hypotheses generated by models. And so how do we compare models in hypothesis space? I think that's an interesting question. One idea is, Marinka touched on this yesterday as well, is sort of having these kind of really huge sponsored unified knowledge graphs. That's a pretty interesting approach where literature is encoded in the knowledge graphs, but you can also take models, extract hypotheses like the ones I showed you, embed those in knowledge graphs too. And if literature and models are embedded in knowledge graphs, you could imagine performing automated comparison, reconciliation, benchmarking of models in the knowledge graph hypothesis space. You can compare the models' knowledge graphs, literature knowledge graphs to identify supported or novel hypotheses. And these knowledge graphs could really power search and recommendations for enhanced discovery and design. And then I think overall, as a community, we should figure out a better balance between efficient learning approaches and naive scaling kitchen sink models. So that's sort of the few takeaways for the future. And with that, I'll just acknowledge my team. These are the fantastic graduate students who've done all the work and great collaborators and funding sources.